Hello and welcome to the video lecture on experimental design. This is part one, where we will discuss the basic principles of experimental design and also learn a little bit of the vocabulary. Today's objectives are these. By the end of the lesson, you should know the difference between controlled experiments and observational studies. You should know why we use experiments. You should know the three fundamental principles of experimental design. You should be able to identify treatment and control groups. You should be able to identify experimental units, response variables, factors, levels, and treatments. You should be able to recall and identify the three basic experimental designs. And finally, you should know what the placebo effect is and how it can be controlled. You'll be ready to answer all of these questions by the time you have completed both parts 1 and 2 of the video lecture. In review, though, remember all data that we study in statistics has to come from somewhere. There are basically three sources. Data can come from observational studies. Those studies can be either a census, where we look at the whole population, or it can be a sample where we look at part of the population and make inferences about what the rest of the population is like. We've talked about sampling procedures in the previous lesson. Today we'll be looking at experiments. In experiments, conditions are imposed so that responses can be observed. In observational studies, we take care not to disturb what is already there. So the essential difference between experiments and observations is this. In experiments, we are imposing conditions so that the responses can be observed. Now, why do we use experiments? The reason is simple. When we're doing observational studies, association between variables is not the same as cause and effect. Experiments can conclude cause and effect, but observational studies cannot. The reason is that in experiments, we can control for lurking variables. By the principle of control, we mean this. Observational studies cannot control lurking variables, that is, variables that have influences on the outcome but are not studied. Consider these two situations and see if you can identify the lurking variables. In one study, elementary students with larger feet were found to have better reading comprehension test scores. So do big feet cause better reading skills? It's also been shown that more shark attacks occur in Florida whenever ice cream sales rise. So does that mean that the sale of ice cream causes shark attacks in Florida? Well, we don't have a cause and effect relationship in either one of these. In the case of the elementary students, the older students are more mature and have had more instruction, therefore they read better. Their feet have also had longer to grow. In the case of shark attacks in Florida, whenever the weather is warm, people are outside, in the water, and buying ice cream. Those two go together, but no one would conceive that one causes the other. In the case of the elementary students, it is the age or perhaps number of years of instruction that is the lurking variable. In the case of the shark attacks in Florida, the weather conditions are the lurking variable. These are known as lurking variables, variables that have an important effect on the outcome but are not considered. Now, why does control matter? Well, all significant variables can't always be anticipated. And this is not always a matter of carelessness. Several years ago, for instance, an observational study found that people who drink coffee were significantly more likely to suffer a heart attack. At first, this was considered to be good evidence that coffee drinking could have negative health impact. Indeed, there was a plausible explanation. Caffeine does affect the central nervous system. It also affects the circulatory system. Blood pressure rises, heart rate increases. These presumably could have an impact on long-term heart health. It was later discovered, though, that in the general population, coffee drinkers are also more likely to be smokers. Now, the original study did not anticipate this effect, and so it was not considered as a variable in that study. So was the effect that was noticed, that coffee drinkers are somewhat more likely to have heart attacks, was that effect due to the coffee, to tobacco use, or maybe something else? Well, no one can say for sure, because not every conceivable variable was recorded, and this was an observational study rather than an experiment. Now, there are three keys to proper experimental design. They are, first of all, control. Two or more treatments should always be compared. We don't simply impose one set of conditions and observe what happens, but rather we have at least two groups with two different conditions, and it's the difference between them that we are looking for. Secondly is randomization. Since we have two or more groups, our experimental units, we call them subjects in the case of people, are randomly assigned into the two groups. This is to avoid unintentional selection bias. That is, a bias that comes from who or what goes into the two groups. Finally, replication is the third principle of experimental design. Replication means that we should have large numbers involved in our study, as large as is reasonably practical. This means that, first of all, the group should be very similar on average. Though there may be individual differences between the subjects or experimental units, on the whole, the two groups should have a similar distribution of those differences. It also increases our ability to detect the differences between the two groups. Variations, say, in means are reduced as the size of the sample that we're working with increases. Now here's some vocabulary we'll need to know as we talk about experiments. The response variable, first of all, is the characteristic of the experimental outcome that is being measured and observed. The response variable is the characteristic of the outcome that is being measured or observed. So, for instance, if we were trying to study ways to increase the speed of 100-yard dash runners, we would be measuring how quickly they can run that race. If we're trying to increase the yield of crops, we'd be measuring the number of pounds or bushels of crops that are being produced. Secondly is factor. A factor is a variable whose effect on the response variable is of interest in the experiment. In the case of an agricultural experiment, for instance, we might be looking at the effect of different amounts of irrigation on a crop or different amounts of fertilizer. The levels are the possible values of the factor. In the case of the study on crops, for instance, we might be putting different number of pounds of nitrogen on our corn crop. Treatments, finally, are each experimental condition. For one-factor experiments, the treatments are the levels, but in general there may be multiple factors, and different levels for each of these factors can be combined to give each possible treatment. This is easiest to see in an example. Suppose that we were interested in the effect of aging wine when it is exposed to sound waves. In one case, we have a vat of wine that's kept in silence. In the other, it has a submerged speaker playing music. The quality of the wine in each vat is to be evaluated after several months. Let's see if we can identify the response variables, the factors, the levels, and the treatments. In this case, the response variable is the quality of the taste of the wine. The factor, there's only one, that is whether or not sound is played into the wine vat. The levels, there are only two levels. Either there is silence or there is sound. The treatments then are simply this: silence and sound. The levels are the treatments in this case because there is only one factor, that of sound or not. Here's another example. Researchers at the Boyce-Thompson Southwestern Arboretum investigated a method of retaining moisture in the soil to promote the growth of the golden torch cacti, which are used commercially in landscaping. They measured the effect of adding a hydrophilic polymer and of different levels of irrigation on the weight gain of the plant. The polymer was either put into the soil or not. The hope was that the polymer would retain moisture and help the plant to grow. Each plant then received either no irrigation, light, medium, heavy, or very heavy levels of irrigation. Presumably, of course, under the presence of more irrigation, the benefit of the polymer would be relatively small. On the other hand, if the polymer did help to retain moisture in the soil, perhaps these plants could be grown effectively with lighter amounts of irrigation. So let's try to identify the experimental units, the response variable, the factors, the levels, and the treatments. In this case, the experimental units are the individual cactus plants. These are what were measured at the beginning and end of the experiment. Secondly, for the response variable, that's simply the weight gain of each plant. The factors were these. Either polymer was added to the soil or it was not. And a certain amount of irrigation was given to each plant. There were two levels of hydrophilic polymer application. It was either added or it was not, and if it was added, it was always at the same rate. Irrigation, on the other hand, had 5 levels. There was none, light, medium, heavy, or very heavy irrigation. The treatments then allow for 10 possible combinations of factors. There are 5 possibilities for the irrigation level. There are 2 possibilities for the amount of hydrophilic polymer. Here's a chart to try to make that clear. Across the top, we chart the level of irrigation. Across the rows, we chart the level of polymer application. This gives us a total of 10 possibilities, such as heavy irrigation with no polymer or heavy irrigation with the polymer. On the other hand, we could have light irrigation with or without the polymer, and there are 5 such levels for irrigation.